Hey, what's up, everybody? Welcome back to the sanctuary. Looks like we got a new boat. That means that we're entering a new topic today. It's still me, Professor C, and we're going to talk about A and P. Specifically, we're going to talk about the cell membrane today. So let's go for it. Okay, everybody, let's talk about cells now. Finally, we've moved on from atoms and molecules, and we're moving our way up the hierarchy, and we've finally gotten to cells. So let's do a few lectures about cells. Let's start with the basics, of course. Let's do cell theory. Cell theory tells us two things about life. It tells us that living things are made of cells. So if you ask yourself, am I alive? The answer is yes, you are, because you are made of cells. Is the pen or pencil that you might be writing notes with, is that alive? And the answer is no, it's not, because it's not made of cells. So what cell theory tells us is that all life is made of cells. And the second part is that all those cells come from some other cell previously. They don't just poof into existence. So when we talk about living things, cells are going to be the smallest structural and functional unit. So yes, I know we started our hierarchy with atoms and molecules, but when you talk about life, cell is going to be the basic unit of life. So all cells come from other cells. Tells us that the continuity of life has a cellular basis. All right, we'll start with cellular diversity here. Notice in the center we have a stem cell, and you might have heard that term before and not be aware of what it is. A good way to start is to think of it as a baby cell that has no job. And what we have to do is give it a job. Now, the great thing about a stem cell is that it actually knows how to do all the jobs. It just doesn't do anything until we tell it, hey, go do this. So if we need a bone cell, we'll tell it to go be a bone cell. If we need it to be a fat cell or a muscle cell, or you can keep looking around the ring there. The point is there are a lot of different types of cells in the human body, but they all begin as some sort of baby cell, a stem cell, and they get told to become some other type of cell. So how many cells are there? Well, there's at least 200 types, and depending on how picky you want to get, right? But 200 is enough. We, of course, aren't going to study 200 types of cells here, but we are going to look at a lot of them during this lecture. So some of them have different components, some of them are different sizes and shapes, and of course they're all going to have different functions since they vary in structure so differently. Now we can immediately change it up and say, well, what do cells have that are similar then? If they have many differences, are there anything that we can make, general sweeping comments about that covers all of our cellular bases? And the answer is, of course, yes. And we're going to look at three major components that all cells, that humans at least, share. Now, we may have touched on some of these before, so let's make sure we remember these if we've gone over it. The membrane, all right, very good place to start. Sometimes we start in the middle, sometimes we start at the outer edge, but of course the membrane is this flexible outer boundary, and we'll be talking about it quite a lot today. Cytoplasm, all of the gooey, sticky kind of fluid stuff where I'm making this wiggly line in here, all of this business here, mostly watery, but a kind of a soup with a whole bunch of little machines floating in it. And these machines are called organelles. I'm underlining over here, and they're going to be, like I said, the machinery, the cellular machines that are going to do the work for the cell. And then, of course, the ball in the center that we've mentioned before, the place where our DNA is stored when we need instructions how to make proteins. This is called the nucleus. So you may have heard it called the brain of the cell. I like to stay away from that idea, but calling it an integration center, that's okay. This talk specifically will deal with the membrane. So we want to start with the basic components of the cell membrane. And it has a name, and you see it in the title there. It's called the fluid mosaic model. Kind of suggesting fluid, that it flows, like it's not static. It doesn't stay in place like a quilt made of patches would kind of sit there. Imagine if you had a quilt made of patches, and the patches would kind of float around the blanket and you saw different patterns every time you looked at the blanket. This is very similar to what we mean by a fluid. Now, a mosaic is exactly what you might think if you think of artwork that's made of little tiny, maybe pieces of glass or little tiny painted stones or something. And you put all these tiny little pieces together and you step back and you see some larger picture. And that mosaic idea is talking about the many, many components that make up a cell membrane. And you can see it here in this image, these little things that look like teeth. Well, maybe they look like phospholipids. More than teeth, because we've studied those lipids already. But those lipids don't stay there. They can move around, they can trade places, they can float up and down, and sometimes float all around the membrane. So we not only have a mosaic of lipids and proteins that we'll say here, but we have a dynamic fluid mosaic, something that's always changing, something that's always moving around. And of course, functionally, what would it do? Well, the key word is very simply boxed right here. It's a boundary. It's a boundary. It's an almost impenetrable boundary in modern cells, although there are some rules that will govern who gets across that boundary and who doesn't. Let's learn a couple of terms here. I do see, and I'm going to put a PL here for phospholipid, and we have seen them before in previous lectures if you want to look back at the lipids talk, and they are going to form a bilayer, right? Two layers of phospholipids, not only one here, but also phospholipids here, forming a second bilayer. We have to remember a few things though, and I'm sure I'll mention this many times. These heads are polar, therefore they should face a watery surface. Same thing up here, the heads are polar, and they should face a watery surface. Now that would mean the barrier part, this part here in the center, that's the actual part that makes the barrier, is the fat. So these fatty acid tails here in the center of the membrane, that's the part that's very hard to cross. That is, of course, unless you are lipid yourself. And if you are lipid, you can kind of sneak through that membrane if you need to. So there's the, and I'll write it here just to be complete, there's the non-polar tails. And it will remind us again on the next slide that these are the boundary. So we have this setup where ECF, extracellular fluid that's on the outside of a cell, and in most pictures anywhere the ECF is on top of the image. This is not always, but most of the time you see it represented just like this. The ECF is on the outside of the cell, extracellular, and the inner part, what we call the cytoplasm a minute ago, could also be referred to as the ICF, the intra, and I'll spell that right here. It's very important to spell this word right. I-N-T-R-A. Intra, not intercellular, but intracellular fluid, aka the cytoplasm. And this is the function of the membrane, to separate the ECF, the extracellular fluid, from the ICF. And these two areas, the outside water and the inside water, will have much different chemistry, and we want that to be kept that way homeostatically, because if we get the cells in the same equilibrium as the environment, then the cell will die. Okay, let's talk about the components of the membrane. The main component is lipid. Hands down, that's always the answer, is it's lipid. Now, most of them are phospholipids, like we just saw, and we see them here in this picture. I'll go ahead and draw a arrow to a phospholipid. We see again its polar head, and it's two non-polar tails, right? And there are the lipids, phospholipids, forming a lipid bilayer, two rows of lipids. So there's the phospholipids, and as it says there up in the corner, 75% of membrane lipids are these phospholipids. So that's kind of the default answer when you're asked what's the primary component of a cell membrane. Phospholipids should be the first thing out of your mouth. Remember, the heads are polar, tails are nonpolar. I'm making this point repeatedly because it's quite important. 20%, the other, missing 25, most of it is cholesterol. So we've made the point that when you see the term sterol, that you think steroid, and we remember that steroids are a type of lipid. So cholesterol usually looks something like this. Although that's not the best way I would draw it. That's the way that this image is drawn it. Cholesterol, kind of this green snaky thing. The point is it's fat, so it's going to hang out with those tails those fatty acid tails, those nonpolar tails. So you should see cholesterol hanging out with the phospholipids inside of the membrane. So most of the membrane lipids are phospholipids. The other large portion is cholesterol. Okay, so let's look at some glyco structures. Now again, we've seen that root glyco meaning sugar, and we can spot them fairly easily if we can look for not a tail inside the membrane, but kind of a tail poking out. And you see it here too, another one, kind of like a chain of hexagons snaking out of the surface of the membrane. These are going to be sugars. So if that tail of sugars is grabbing onto a phospholipid, we'd call that a glycolipid. However, if that tail of sugars is hanging on to a protein, which is what the purple things are, so we could call it a glycoprotein. So glycolipids and glycoproteins make a small component of the membrane, and they're used for things that have to do with immunology, kind of like acting as some sort of an ID, so that when defensive cells come up to a cell and says, "Show me your ID," they can say, "Hey, here it is, please don't kill me," or send the other cells out to attack me, right? So other things that these glycoproteins and glycolipids may do is stabilize the membrane and assist in some kinds of enzymatic reactions. All right, now the proteins. This is the second most common thing. So if lipids, mainly phospholipids, were the primary component, protein will be the secondary component to a cell membrane. So three major types of these purple blobs. Some of them act as transporters or channels, and I can put a box here showing you an example of one of those. You can see just by the way that it's designed on this image that things could potentially go in and out of this and pass that barrier. That's that fatty barrier that we talked about earlier. Okay, receiving outside information. That sounds very suspicious, like we've heard that before. Those are what receptors do, right? So maybe, and I'm just going to draw this here, maybe this is one of those Y-shaped structures that I always draw in my pictures of a receptor sticking out. It's calling that a globular protein. Okay, that's a very kind of generic thing to call it, but it could be a receptor protein receiving information from beyond the cell. And there could be, if you see any threads in a picture, and I'll sketch these in real quick, down here in the ICF, the intracellular fluid, again the ECF would be the fluid on the outside of the cell. So here on the inside of the cell you could see these fibers running through the cytoplasm. These are called threads of the cytoskeleton, and I don't go much into depth about it, but I will show you a slide during the organelles talk about the cytoskeleton. And it does what you would think it does. It acts as kind of a skeleton of the cell. Some of these proteins that are on the inside here can grab onto these cytoskeletal elements and use them as anchors. So up here when we say the three major roles of these membrane proteins are channels or tunnels for transport, receptors for getting information, and some act as anchors to attach to the cytoskeleton, kind of hold the membrane onto the cell. Some proteins, as we said, act as receptors. And again, they're not ever drawn as a Y-shaped thing on images that artists draw, but I like to add them when I'm taking notes, something like that. There's a receptor. It's waiting for something to bind to it. We don't care that it's called a ligand, although it may be under some circumstances. The point is, it's picking up information, the receptor is, from the outside world. We haven't really talked much about what happens, except in the instance of a homeostatic control mechanism. We said the receptor may send a signal to somebody else, and that is indeed what's going to happen here. Now we can sum up all this rigmarole. I know it kind of looks a little overwhelming when you first see it, but let's just call it, for better or worse, magic. Okay, some sort of cellular magic will occur. And this is the idea of proteins as receptors. So something binds, like maybe it's a neurotransmitter from the nervous system or a hormone from the endocrine system, and it binds to the receptor and causes this G protein cascade is the fancy word for it, which is, a bunch of magical chemistry that will happen. And the cell will do something in response to receiving this signal. So this is proteins as receptors. Okay, as I mentioned, sometimes you may see threads down here, the cytoskeletal elements, the long protein threads that would be inside of the intracellular fluid or the cytoplasm. And you may see some of these proteins here grabbing on to these threads. Now we have seen a word here that I just want to bring our attention to. Even though we didn't mention it, there's these two types that I've seen on these images, integral and peripheral. Just take a look at what they look like. An integral protein is going to be integrated all the way through the membrane. It goes from one side to the other. Now you could argue here this channel protein is integral, and I would argue that too, since it goes from one side to another. Now this down here, the one that's on the outside or inside of the cell membrane, it does not go all the way through. We would call this a peri-peripheral. Peri meaning around. So it's around the edge, but it doesn't quite go through the membrane like an integral protein was. Okay, we can practice labeling real quick if you would like to try this. First of all, just take a look around the image. Some things should look immediately familiar. Some things you may have to think through. If you'd like to try it yourself, go ahead and pause the video. But I'm going to go ahead over the answers right about now. Okay, so A, well, I see a blue thing. Sometimes they're purple, sometimes they're blue, stuck in the membrane. It's going all the way across. I would definitely call this an integral protein. And probably I would call it a transport protein or a channel protein, because it looks like something can go through there. I can cheat and look forward at I. I looks like an integral protein too. I'll just put integ-per for short. Again, it doesn't look like a channel protein. It probably has some other function, but it is integrating the membrane. It is going all the way through. So I would call A and I both integral proteins. B, I see that it does kind of poke down into the membrane, but doesn't go all the way through. So I wouldn't call it integral. I would start by calling it peripheral. I'll just put peri, a periprotein, a peripheral protein. It looks kind of globular. Maybe we could get away with just calling it a globular protein. Maybe it's acting as a receptor. You can't really tell just by looking. But it's not integral, so I would call it a peripheral. So would I call H peripheral. Right? It looks like it's not quite all the way through the membrane, but it is poking into the membrane a little bit. So perhaps that's acting as some sort of anchor holding on to these threads of cytoskeleton, which is what J would be. If we're going to jump ahead a little bit, I'll just put cytoskeleton. Those are the threads of the cytoskeleton. All right, let's head back up to the top of the image and look at C. So C, again, I know it's a protein of some kind, but I don't think they're looking for, peripheral or integral here, because I see the little ribbon of hexagons sticking up out of it. And I know that little ribbon of hexagons is a carbohydrate. So, in fact, if I look at D right here, they're asking only about this little string of hexagons. So that's a carbohydrate. That's just a carbohydrate. That's a sugar. And because it's attached to that protein, this would be a glycoprotein, having something to do with cell recognition, ID, enzymatic reaction, a variety of function. Okay, and then just to get the top row clear, E all the way on the right over here. E looks like it's pointing to two of these balls, and we know those are the heads of the phospholipids. So I'm just going to cheat real quick and put heads, and we know those are the polar heads of the phospholipids. And then down below M is pointing to the tails, right? So that's the fatty acid tails, non-polar fatty acid tails of the phospholipids. Okay, well, what are we missing then? Not much. Let's head all the way over to the left and focus on F now. Letter F. Okay, it looks like some hexagons, but they're not poking out. They're poking in, right? They're poking into the membrane. They're hanging out with the fatty acids. So it must be some kind of a lipid. And I know it's not a phospholipid, therefore it must be cholesterol. So I'll just put chole right there, cholesterol. G, looks like it's pointing to a phospholipid, but the head's gone. The tails are still there, but then we have this string of sugary stuff coming out, another carb. So that's half carb, half lipid, so let's just call it glycolipid. Beautiful. Okay, sliding on over to K. Yeah, and I would be tempted to call that a peripheral protein because it's not spanning the membrane. It's certainly not integral. It's not even really a globular protein. It's kind of a weird flat guy. You could get away with calling that a surface protein, maybe. I would call it a peripheral again, though, if that's my only option. A peripheral protein. Some books probably do call that something like a surface protein. What's it doing down there? Maybe anchoring to the cytoskeletal threads also. L, we didn't mention in our talk, but we have seen this, protein that's twisted into spirals. If you've watched my video on protein structure, that's one of those secondary structures of protein called the alpha helix, where it's twisted into a spiral. So there's an alpha helix protein, probably more important to us. It spans the membrane, therefore it is an integral protein. All right, how'd you do? Pretty good? Keep trying if not. Just takes practice. Thanks for watching the video. If you enjoyed it, got something out of it, check out some of the other videos in the series. See you for the next one. Bye-bye.